This paper discusses length scale dependent elasticity in DNA. It starts with a simple toy model, which shares many of the features of the DNA mechanical model discussed further on. This is a one-dimensional elastic chain with two types of springs, one connecting nearest neighbors with stiffness K, and the other connecting next nearest neighbors with stiffness K prime. The system is in thermal equilibrium and the distances between the masses fluctuate. Now, these fluctuations will be governed by effective stiffnesses K1, K2, K3, and so on. If K prime is different from 0, then K1, K2, K3 are all different. In other words, the elasticity is length scale dependent. In particular, if K prime is positive, we find that K1 is smaller than K2, which is smaller than K3, and so on. Which means that the chain is softer at short length scales and becomes stiffer at longer length scales. If K prime is negative, you obtain the opposite result. See details in the paper. Note that K1 is not equal to K. A convenient way to solve this problem is to use Fourier transforms because the Fourier modes are uncoupled. The Q mode has a stiffness K tilde Q, shown in figure B1. The asymptotic stiffness of the chain, which we call K infinity, is equal to the momentum space stiffness K tilde at Q equal to 0. The local stiffness K1 is the harmonic mean of K tilde. Hence, it contains contribution from all Q's. As one increases the length scale, smaller and smaller ranges of Q contribute to Km, until at infinite length only Q equal to 0 matters. A plot of K tilde versus Q gives a direct and convenient representation of how the system behaves. A strong dependence of K tilde on Q indicates strong differences between local and asymptotic stiffnesses. This is a consequence of strong coupling between nearest neighbors. Conversely, a weak variation with Q indicates that elasticity is weakly dependent on the length scale and that there are weak couplings between nearest neighbors. Thus far, we discussed the case of two couplings, K and K prime, but all these can be generalized to a more complex situation, including further neighbors' interactions. Now we turn to the mechanical model of DNA. The 1 degree of freedom we had before is now replaced by 3 rotational degrees of freedom, 2 associated to bending, tilt and roll, and 1 to twist. Again, the momentum space representation can be used to introduce couplings to further neighbors. This momentum space stiffness will be described by a 3 by 3 matrix, instead by a single function K tilde Q. As in the toy model, we will have length scale dependent elasticity, characterized in DNA by length scale dependent torsional and bending persistence lengths. The expressions for the persistence length strongly resemble those for the stiffness Km of the toy model. Details of the analytical calculations are in appendix. We have performed molecular dynamics simulations to calculate the stiffness matrix in momentum space. Figures 4 and 5 show the results for oxDNA, a coarse-grained DNA model, and for all atom simulations. The stiffness matrix has non-zero off-diagonal elements, which we discuss in the text, but we focus here on the diagonal stiffnesses. AT and AR are tilt and roll stiffnesses, bending modes, while C is the twist stiffness. Both coarse-grained and all-atom simulations show that the roll stiffness is weakly dependent on Q, while tilt and twist stiffnesses are strongly dependent on Q. As a consequence, the bending persistence length is only very weakly length dependent, because the analysis shows that this quantity is predominantly determined by the roll stiffness. The torsional persistence length, on the other hand, is strongly length scale dependent, as it is predominantly determined by C tilde, the twist stiffness. In the all-atom case, the effect is even more pronounced. In particular, the torsional persistence length divided by 2 varies between 50 to 130 nanometers. This variation is fully consistent with the experimental data. Magnetic tweezers, where length scales of the kilobase pair range are probed, give values of 110-120 nanometers, while measurements from local fluctuations using, for instance, fluorescent probes systematically give much lower values, around 60 to 70 nanometers, but even lower. Summarizing, we have shown that the couplings in DNA extend beyond nearest neighbors, and that these are responsible for length scale dependent elasticity. There are remarkable differences between different degrees of freedom. Non-local couplings are very strong in the case of tilt and twist, but very weak for roll. The consequences are that the bending persistence length varies weakly with the length scale, while the torsional persistence length is strongly varying with the distance, which is fully consistent with experiments.